Welcome everyone to today's webinar episode that is being put onto you by Techcyte. We're very happy to be with you today. A few bit of housekeeping items. We encourage any questions that you might have, and for that, you can use the chat function in your browser. And without further ado then, let's get started. My name is Troy Bankhead. I'm the Director of Techcyte Europe. I'm actually located in Europe, and I'm the VP of Marketing. And we have a very special guest today. His name is Dr. Larry Vaughan, who is the Director of Scientific Affairs and Diagnostics Solutions with BD. And he will be addressing the current and future perspectives with cervical cancer screening, so the Pap smear, and the associated technologies with it. Today in the webinar, we're going to start off with just a short bit about who Techcyte is, and then we'll get straight to the meat, which is the current state of the science, where we're at, the workforce issues that I'm sure you're all going through, and the digitization of cervical cytology. And at the end, we'll have a short Q&A session to be able to address any questions you might have. So first of all, Techcyte's been around since 2013. We are a technology company, and we actually have three verticals: human, veterinarian, and environmental. In the human space, though, which is our, of course, main focus, we are now active in the AP side of the lab as well as the CP side. Traditionally, we've been in the liquid mediums, the cytology mediums of the laboratory, but thanks to a relationship and collaboration that we have with Mayo Clinic, we are now developing solutions in the histology side. We, as a company, are based in Utah in the United States. We're currently well over 130 employees, and we also have an office located in Luxembourg. And as a company, you might think of Techcyte as AI, but we are much more than that. Of course, you'll notice that data curation and the AI algorithms are one important component of who we are, but we also devote enormous resources to developing and building out the workflows so that they're efficient and pragmatic in the laboratory environment. While we don't make scanners, we have been we've had to get really good at image acquisition as well, and so we consult with any parties who want to work with us and are able to recommend appropriate scanners for different situations. We are able to be global. We are based on AWS, and security and scalability are of foremost concerns for us, as well as compliance, whether it be FDA, CE marking, HIPAA, GDPR, ISO, SOC 2. We have an entire department that does nothing but that. And then, of course, getting information from our system into the laboratory systems is of utmost importance to us as well, and we have skill sets there as well. Overall, Techcyte as a mission wants to positively impact the health of humans, animals, and the environment through the use of artificial intelligence. That's in a nutshell who we are as Techcyte, and right now what I'd like to do is turn the mic over to our special guest, Larry Vaughan, who is the Director of Scientific Affairs at BD. Larry?
So much, Troy, and thanks everybody. I'd like to add from a BD perspective, my welcome to you and thanks for your time today. What I wanted to do is really just set the stage for the discussion of the technology and talk about current and future perspectives of how we see the world of cytology. So in terms of current state of the science, and this is an attempt just to put the history of the PAP and HPV screening onto one slide, which is the challenge, as you know, but I think we're all aware that George Papanicolaou invented the PAP way back in 1928, and it really didn't have any traction until he published in the 40s with some noted OBGYNs of the day. And then gradually he began to be taken seriously and physicians began to be trained. And by the 60s, it was really in large scale implementation. And this really led to a large decrease in both incidence of disease and mortality due to cervical cancer. And that went along for about 30 years or so globally until around the 90s when we had the introduction of liquid-based cytology. And this really did a couple of things, and it took off and was really supplanted in developing countries the conventional PAP. The conventional PAP, a good conventional PAP is a good PAP. However, it's a one-up, individual smear, and it varies in quality in terms of drying artifacts and mucus and blood and so forth. So the advent of liquid-based thin layer, so-called thin layer cytology, really, first of all, added quality control to that and how the cells are deposited on the slide. It also allowed us to automate the whole process, which allowed us to scale. So that was an important development in the 90s for both ThinPrep and SurePath. And then added to that in the 2000s, we had the advent of HPV testing. And for the most part, that has been gone along so-called co-testing, where you're doing both PAP and HPV from the same sample. And again, the liquid-based PAP enabled that because you had residual material that you could run an HPV test on. So that has become the norm, and particularly in the US, the co-testing is still a predominant mode of testing. However, in more recent times, we've seen due to the large body of data that has accumulated, that of course HPV is more sensitive than PAP for detecting disease. So that has now started to take over and do primary screenings. We're moving from co-testing HPV plus PAP to HPV then PAP. In other words, we're triaging HPV positive results from a primary HPV test. And added to that, and it's exciting, in the 2020s, and we were one of the first PCR tests to get CE mark approval for self-collection at home. And now that's starting to really take off ex-US, and we're hoping this year we'll start a trial. The SHIP trial will start very shortly to bring that to the United States. So what we see now is we're adding self-collection as another means, particularly to address the underserved women. Women, roughly in the US, 30% of women do not participate in screening, and that they account for 60% of cancers. That's a serious problem. In order to move the needle, we really need to get after the underserved. And I would add that since the initial success of the PAP in bringing down the incidence, we've plateaued over the last 10 years or so for squamous cancers, and over the last 40 for adenocarcinomas. So we really have to do something different to move the needle to reduce mortality and morbidity. So if we take a look across the globe, this is a recent review from the last few years ago, although of course it's out of date already. This is Sylvia de San Jose and colleagues, and you can see across the globe who's doing what in terms of screening. And of course, the immediate thing you'll notice in terms of landmass, cytology is still a predominant tool. We have a mixture then of HPV plus cytology, some DIA, nothing at all in some cases. But we see that advent of primary HPV in places like Australia and Nordic countries in particular. And if you look at numbers, you can see that, again, cytology is roughly 3/4 of all countries doing testing. But BIA is on the decline. I think it's not a very sensitive or reproducible test. Of course, we have the march of primary HPV, as I mentioned, and that's particularly the case in higher income countries. So, as I've said, we're moving from this transition away from the qualitative morphological description of the disease, if you will, to looking at the causative agent, HPV itself. As I've said, it's roughly twice as sensitive as a PAP. So there's good reason to go there. However, we don't see the PAP going away, and why there's a need for new technology. First of all, as I've said, it's a major player in the lower middle income countries, where we see its predominant tool used to do primary screening. And also, even in a developed situation where we have primary HPV, we're still using cytology for the most part as one of the most popular triage methods. So you get that benefit of the added specificity to make sure you're not over-referring women to colposcopy who don't need it. We've also, of course, have extended genotyping, which we'll talk about in a minute, and we have things like P16, dual stain, or Roche Syntec Plus, and methylation products are starting to come into the market. So we have a number of options, but cytology is still a predominant player in terms of triage as well. So we have a need going forward for cytology. And just as a reminder, this is just some data from our baseline trial, but just to give you an idea of numbers here, to reemphasize, we're screening for precancer, not cancer. We want to eliminate at the precancer stage. That's where the intervention is. And roughly speaking, 15% of folks would be positive. We cannot, of course, send those to colposcopy. We need a triage method. And you can see the numbers of SIN3 and even numbers of cancer are manyfold reduced from the number that we have a positive. So a positive HPV is not necessarily the problem. Persistent HPV infection leading to SIN3 is the problem. That's where we need to intervene and do either a biopsy or a treatment at that point. And again, we want to catch it before it becomes a cancer. The goal is not to screen for cancer. So this is a study we did publish last year, which kind of illustrates some of the challenges here and also some of the opportunities. If you look from left to right here, we see that, you know, the genotypes are really what's driving disease. This is baseline SYN3 risk, and you can see this stair-step reduction in risk regardless of cytology. So you can see the high grade or ascocellulose or normal, you see the same profile here, obviously with reduced risk as you go down the abnormality scale here. But what this tells us is that we can really do triage with extended genotyping, but that also cytology can play a role here as well. So let's take a look at that. This is a receiver operating characteristic or ROC curve plotting sensitivity of these data versus 1 minus specificity. And as you can see in these curves, you want to be pushing into the top left, which is maximal sensitivity and specificity. A couple of points to note here. First of all, we see the various options here as the data points on the curve. We've also plotted the cytology result from the OnClarity trial, and superimposed on it, we've also added the IMPACT trial, which is another trial done shortly thereafter. It validated dual stain product, and we're just plotting that for comparison purposes. These trials are very similar in design, very similar in their demographics, positivity and vaccine status. So it's appropriate. There are about 35,000 women in both trials. So it's a lot of data here to look at. So we can see here that, you know, first of all, combining extended genotyping and cytology offers some benefits over cytology. You're increasing sensitivity. Also, dual stain seems to sit pretty nicely on this curve. And as part of this genesis, we were asked, well, what about if you just did the same analysis without using cytology? And the bottom curve here in this next plot, you can see the dotted line. That's just, as you can see, just straight up extended genotyping, taking these various combinations of genotypes and seeing where you might refer people to colposcopy or not. And you can again see that you see the cytology from both trials. And I should mention that cytology here is not the same in both trials. One is better than the other. And it just shows you that the reproducibility of cytology, even in a controlled trial situation, is not what you would like it to be. And the molecular testing is much more aligned and reproducible. So the point take home from this is that if you combine extended genotyping and cytology, you can improve triage, right? And we'll take a look at some numbers in a second. So we have this ability to use the modern extended genotyping and the older PAP to actually improve the power of two, as it were, to improve performance. So we're seeing a push into that top left corner, meaning we're getting better overall triage when combining these two technologies. And just to bring this home, these are a lot of numbers on here, but what we're doing here is actually looking at the cumulative sensitivity, specificity, and really what I want to focus on here in the interest of time is just the colposcopic efficiency. In other words, how many colpos do I have to run or to detect the SYN3? And this ratio, the lower number, is obviously better. And you can see here from the IMPACT trial, we have similar data. We bracket that with a number of these triage options that we have with extended genotyping, with or without cytology. So we have options here. The other thing that's nice from a global perspective is we have different risk thresholds in Europe versus the US, and you could tailor your cutoff versus a non-off switch. You can decide where you want individual guideline committees could decide where they want to make that cut and refer women to colposcopy. And I should mention that we'll get more guidance on this from the updated ASCCP app, which we expect to happen in the next few months, where we'll see guidance for both dual stain and extended genotyping into that app that will guide us from a US perspective. So I don't need to tell you, if you're working in this field, that we have a challenge. We'd like to use cytology. I just made the case for that. But what about the workforce? It's rapidly declining. Most cytotechs, or I'll say not all, but certainly a lot of them are moving to retirement age. They're not being backfilled, as we see here from the numbers. And also the cytopathology, there's starting to see some pressure on that as well. For cytopathology, fellowships are harder to fill. And I would sum all of this up with a simple statement from a European pathologist I talked to last fall. She said, look, I used to have 6 cytotechs, now I have 3. Essentially, hurry up with this technology because we have a need for it in the field. And I think you know that already. And so where do we go from here? We've got all these challenges. We've got these movements and guidelines. We have the world is changing and adapting to new screening guidelines and technology that's coming on. We still have a place for cytology for sure. It's a large player across the globe. We have a labor force challenge. So what do we do? So clearly AI and AI-guided screening can alleviate some of that pressure, and that's what we're here to talk about today. So we can certainly bring some benefits with remote screening, workflow efficiencies, digital storage, and LIS integration. So we can alleviate some of the pressures that we're under, and certainly from a labor force perspective, by making screening more efficient. So that's what we're here to discuss today, and I'll let Troy come back in a second. But so we're moving from this glass slide review with manual or guided microscopy to a digital image that can be stored, sent across the globe or across the hall to a reviewer, and you can consult remotely from your home and whatnot. So with that, I'll hand it off back to Troy to add a little bit more meat to the bones in terms of the product offering. Thanks, Troy.
Thanks, Larry. In fact, with that intro and with that insight into the current state of cervical cancer screening, I wanted to just give you an overview of the technology that we use and the software that we've developed. What we've created is a workflow based on an algorithm, of course, that is tailored to cervical cytology reading. It supports both ThinPrep and SurePath slides, and it works with several scanners that we validated to date. And as time goes on and we are presented with the opportunity to validate with more scanners, we will continue doing that. Now, our software is geared around the cytotechnologist or the cytologist. And the goal is to present the most suspicious objects or organisms first in a list of Bethesda classifications that help the operator to be able to eliminate the time spent searching for objects on a slide and rather spend their time analyzing and confirming what the AI shows them. Our software, as I mentioned, is fully Bethesda compliant. And recently in a study that we ran at a hospital in Mexico, we reviewed 300 negative slides that the cytologists there had deemed no further review. We ran those slides through our software, and the same cytologists were able to find 17 low-grade cancer cases, which is indicative of the results that our software brings. It's not just time savings, it's not just better working conditions, but the software does it tends to be more consistent and accurate than humans. This being said, the workflow that we use involves the cytotechnologist. The creation of the slide happens using industry best practice liquid-based cytology, as I mentioned, either SurePath or ThinPrep. Once these slides are prepared, they are scanned. We require 40x magnification, and the entire dot or the entire sample is scanned and uploaded to our servers. Once it's there, we perform deep machine learning on the sample, and it just takes a few minutes. And the results are then presented in the interface that I just showed you, where the cytotech reviews the images. They build their case, looking through squamous glandular. We even do other organisms, a few STIs, and things like blood cells, yeast, stuff like that. And they then are able to build their report and send that report to the LIS. The way our system is conceived, therefore, brings several benefits into the laboratory. It increases accuracy and efficiency. It helps address the pretty much universal shortage or difficulty of hiring and retaining qualified cytotechs. It enables either remote reading or remote scanning in a centralized location because everything is web-based. And it can have possible or potential use as a primary or secondary screening tool, which increases the efficiency of laboratories. Because there's no equipment other than the scanner, it utilizes lab space a lot better. And of course, while we do have PDF report generation, we also use an open API, which enables any LAS system to come and retrieve any data from our system, including images, and insert them into the laboratory's LAS. I'd like to just briefly go over as well the relationship between Techcyte and BD. Some of you might have seen the announcement that we recently made, but today BD is now the worldwide exclusive distributor of the Techcyte cervical cytology solution. That coincides with BD's announcement of their retirement of one of their flagship products, which is Focal Point. And we are starting out the collaboration by presenting it or bringing it to the European market, because we already have the CE clearance in Europe for our cervical cytology solution. However, we are currently pursuing our FDA clearance that will enable us to expand our commercialization to the US. And with that, Larry, maybe I'd like to turn the time back over to you, now that I've given our visitors a bit of an overview of our technology, just to wrap up today's webinar and move into the Q&A.
Yeah, thanks so much, Troy. Really not much to add here. I think we're very excited that we're on the precipice of bringing this to market, and I think it offers a number of benefits, as Troy pointed out. But I'd love to get your feedback. And I don't know, Troy, I think you're managing the Q&A from your side. Do you have any questions at this time? We have a few minutes certainly for questions. Oh yes, I'm sorry, I do have one slide. I'm working off script here, but so just to close out, I think I've said most of this. Cervical cancer screening is changing. There's a global shift from primary cytology to primary HPV. We've seen that we really do see that self-collection is going to transform how we screen. It's going to be an adjunct to the physician-collected sample, and it's becoming very popular in countries where it's been adopted already. We do have that cytopathology workforce problem that we're addressing with AI and improving efficiencies. And in the near future, I think we're going to see, remote or at-home self-collection, in drop-in clinics, whatever. We have to maintain the ability to triage HPV-positive cases, be they from a primary self-collected sample or a physician-collected sample. And we do believe that digital cytology and extended genotyping really bring a lot to the party here. So I'd love to get your feedback on that. I'm sorry, I forgot to cover that slide.
No problem, thanks. Okay, let's then go ahead and move into the Q&A portion of today's webinar. We have a question here from Tiffany, who says, our lab has both ThinPrep and SurePath sample types. If we use Techcyte BD, would we still need a Hologic or another viewing system for ThinPrep? And so I can actually address that, Larry. In fact, that's a good question. When I say that our system is compatible with SurePath and ThinPrep, I do mean just the sample preparation. And so once the samples are prepared, you don't need any other viewer or any other software. We can use those slides as is in our reader. Jonathan asks, how efficiently can Cytotechs get through these cases using AI? Another good question. We are currently running more qualitative studies on the actual time savings. I can give you some anecdotal examples of some of our other verticals, such as parasitology, where Mayo Clinic has been able to triple their capacity, and ARUP in the US has been able to reduce a 3 to 5 minute read time down to around 15 seconds, and that does include back reads. Now, with cervical cytology, the time savings are similar, although we don't have exact numbers today because our testing up until now has been focused on the technical and performance aspects of the software. But the time savings that we've seen in all of our trials and in the labs that are using our system today do reflect the same scales of time savings. And that's because the time savings comes from not having to spend all their time looking for the objects. Here, they simply view the objects that the AI proposes, and then either confirm or challenge what the system has shown them. So we've got a question from Rose, who asks, are there any updates to the daily case limits that cytotechs can do if they use this system? Do those limits still apply for now? This is another good question. The limits that laboratories place on the daily cases that are able to be reviewed have to be part of that laboratory's quality assurance systems that are in place. What that means is laboratories, once they start using our system, have to compare it with the reliability of the existing standard that they are using. Once that is done, that frees the lab up to then review the quantities or the limitations that they put on the number of cases reviewed per day by cytotechs. And so that's a very laboratory-specific decision that needs to be made. But the short answer is, yes, they can be increased, but it's up to every lab to determine, according to the cytotechs that work there, their experience, how comfortable they are with it, in where they set those limits. We've got a question from Esther that says, how did your platform perform with regards to glandular lesions? In all of the cases, or all of the organisms that our software is able to detect and classify, we set thresholds and we set performance metrics before we release the product. And basically, while I guess the question is does it work? Yes, it does allow for cytotechs to be able to view and detect things like glandular lesions. I guess I hope that answers that question, but it does allow to screen for those. Bob has asked us, do you have any online AI-assisted case to practice? Interesting. The way that we currently operate when we meet new laboratories is to schedule a demo with them. We show them the software. They usually invite the medical teams, and we're able to introduce them to the software. After that, the next step is generally actually to bring them on board, give them access to our environment, and let them practice on cases. So everything we do is online, and we can most definitely, if you're interested, just reach out to us, and we can organize both the demo and let you and your teams go in the system, perform test cases in order to practice. We also have training that's online that is part of the software that can help with education or certification or even ongoing education. We've got a question from Jerome, who asks, how would we decide which scanner to use? It looked like you have a lot of scanners that could work for your software. And that as well, man, we would love if there was a magic bullet solution of one scanner that could do everything. But there are a few factors that impact that in terms of making that a reality. The first is that we can't recommend a high volume scanner to a small laboratory. It would be just massive overkill and blow their budget. Likewise, we're not going to propose a super small scanner to a high volume throughput laboratory. And so there's the notion of volume that comes into play. Aside from the volume is the quality and the capability of the actual objectives. We do need a certain magnification with a certain numerical aperture that gives us images that are of good enough quality to be analyzed by the AI. And as such, we need to have that discussion because keep in mind that cervical cytology is one of the things that Techcyte does, and that is the subject of today's webinar, of course, but we also do parasitology. We do a manual differential for blood. We do bacteriology, VV detection. We do bladder cancer detection. And I think I mentioned our fecal ovum parasite detector. And others are in development, including frozen section. And so each of those has their own particularities. And depending on the lab's interest, you can essentially have one scanner that performs several laboratory tests, which of course is advantageous for the ROI of a particular scanner. So to briefly answer that question, There are a number of criteria, file output of the scanner and things like that. Does it load the slides horizontally or vertically? Things like that can impact the choice of scanner that we use, and we work with scanner manufacturers to make those decisions. Sorry, we work with the laboratories to make those decisions. So we've got a question from Giselle who says, hello. So it doesn't matter what kind of staining you're using. It does matter. The stain, of course, has to be one of the papillomacow stains that's approved and used by ThinPrep or Hologic. Question from Brian who asks, are you partnering with any of the cytology schools to help prepare future cytologists to accurately and efficient utilize this technology? Today, no, but that's a wonderful idea. I think that especially with the relationship with BD, that would certainly be something we'd love to look into. As Larry spoke to, Larry, did you want to make a comment?
Yeah, just to add to that, I mean, I think we heard a talk at the ECC Congress last fall where one of the benefits of AI is that you can do training, right? You can train remotely and share cases, and that's going on in places like Korea. And there's a whole national database being set up. So I think that's a very insightful comment and something we'll bring back to the team.
Yeah. And Juna has asked us as well, what is the name of the new digital platform, and has it already been submitted to the FDA? Juna either has some back information or very insightful, because we are branding the platform today. We're not ready to unveil it to the world, but we're very excited about it, because not only will it join the anatomical pathology side of the lab with the clinical pathology side of the lab, but our aim is to overcome a lot of the functional operational silos that exist in the laboratory workflows today. Of course, with tests within the system that are based on AI. And concerning FDA, it's not as easy as just submitting it. We haven't completed our final submission, but we are definitely already underway, both with the clinical studies and with the, from the Q-subs to the various submissions and discussions back and forth with the FDA. So yeah, and sorry, someone said, is correcting me. Thank you very much for that. Yeah, I meant to say that we accept PAP stains that are approved by and used by BD's SurePath or Hologic ThinPrep. And I think I said ThinPrep or Hologic. But they are both, of course, compatible stains and slide mechanisms. But it looks like, actually, we have reached the end of the questions. So without any further ado, Larry, I would like to extend a warm thank you to you for joining us today and helping us introduce this technology to the world. If you want to say some closing remarks.
Thank you.
Wonderful. Great. We would like to thank all of you for attending today's webinar. We will be sending the recording out to you so you can have access to it shortly after the webinar today. Thank you, and have a wonderful rest of your day or evening, depending on where you are in the world. Bye-bye.